Students, this is chapter number 2, Biological Molecules, and in today's lesson we have to see the primary and secondary structure of proteins. Let's see the primary structure first. A polypeptide or protein molecule may contain several hundred amino acids linked into a long chain. The particular amino acids contained in the chain and the sequence in which they are joined is called the primary structure of that protein. If you remember from our previous lesson, I told you and tried to explain you how the amino acids are linked together by peptide bonds. So what primary structure of the protein actually tells us? It tells us the number and the sequence, the particular amino acid and the specific sequence of them in a structure in a specific protein. There are an enormous number of different possible primary structure. Even a change in one amino acid in a chain made up of 1,000 may completely alter the properties of polypeptide or protein. Here, if you can see that different amino acids are linked together to form the primary structure, and if I change only one amino acid, it will lead to total different protein formation. Notice that at one end of the amino acid chain there is an NH3 group, while at the other end there is a COO group. These are known as amino and carboxyl ends respectively, or you can call them as N and C terminals respectively. This animation also is helpful to understand the primary structure of a protein. If you see that in the beginning there were many amino acids which then linked together in a specific sequence to form the primary structure of a protein. This again is showing you how the N and C terminals are already there in the primary structure of the proteins, and you can add up more amino acid if needed into the growing ends, which are N and C terminals. The primary protein structure is the chain of amino acid that makes up the protein. This is the secondary structure now. The amino acids in a polypeptide chain have an effect on each other even if they are not directly next to each other. So now we got to know in the previous slides in the primary structure that amino acids are linked together. So even if they are not directly linked together, in a chain, in a polypeptide chain, one amino acid would be affected by the other. A polypeptide chain or part of it often coils into a corkscrew shape called alpha helix. This is the structure of alpha helix, and if you can see, there is some kind of interaction between the amino acids which are not really directly attached to each other. This secondary structure is due to hydrogen bonding between the oxygen of CO group of one amino acid and the hydrogen of the NH group of the other amino acid, which is 4 places ahead of it. So now we got to know that the interaction between the amino acid is 4 places ahead of it between oxygen and hydrogen atoms of the amino acids. This is the alpha helix, where you can see that the polypeptide chain is kind of coiled together, having hydrogen bonding, which is shown by this blue color. Hydrogen bonding is between the oxygen and hydrogen of different amino acid. These amino acid lie 4 places ahead of each other. If you can see here, this is one amino acid. Its oxygen is interacting with the partial positive charged hydrogen of another amino acid. Each amino acid has an NH and CO group. This is something that we have read in our previous lesson. All these groups are involved in hydrogen bonding in the alpha helix, holding the structure firmly in place or in shape. Hydrogen bonding is a result of polar characteristics of CO and NH group. I told you in our previous lesson that the basic formula or basic structure of amino acid itself has dipoles. The NH group and CO group are the terminals where more amino acid can be attached. Secondary structure also include another structure The amino acid in a polypeptide, this is the same slide that we read just now. This is the beta pleated sheet, which I just told you that there is another structure formed by the secondary structure of proteins. Sometimes the hydrogen bonding can result in a much looser, straighter shape than the alpha helix. So now we got to know that the coiling is not only in alpha helix form, it is also in beta pleated sheet form, which is a loose sheet where the amino acid are linked together loosely with each other. Hydrogen bonds, although strong enough to hold the alpha helix and beta pleated sheet structures in shape, are easily broken by high temperatures and pH changes. If you remember from your knowledge of O levels, we read that denaturation occurs in protein when the temperature and pH is taken beyond the optimum temperature and optimum pH respectively. So now what we got to know that the amino acid in alpha helix and beta pleated sheets are held together by hydrogen bonds which are strong enough to keep them in shape, but with increase in temperature and pH they can easily be broken down, leading to denaturation of proteins. This is the structure of beta pleated sheet, which is another form in which the polypeptide chains can coil upon themselves. These are also shown in animation here. Some proteins or parts of the proteins show no regular arrangement at all. So sometimes it happens that the proteins do not have a very regular shape. It all depends on which R group are present and therefore what interaction occur between amino acid in the chain. If you remember, I told you that there are 20 amino acid. All of them are differing with each other only by this R group. So the arrangement or the structure of protein is also dependent upon this R group, depending upon which amino acid contain what kind of an R group and how it will interact with other amino acids. In diagrams of protein structure, alpha helices can be represented as coils or cylinders, beta sheets as arrows, and random coils as ribbons. Here you can see in this diagram, and this is the structure of lysozyme. Here alpha helices are shown as blue coils, beta sheets are shown as green arrows.